Biochemistry, cellular part. First, we will start from cell cycle phases. Cell cycle phases have checkpoints. I'll go by one. Checkpoints control transitions between phases of cell cycle. This process is regulated by cyclin-dependent kinases, CDKs, and tumor suppressors. M phase, short phase of cell cycle, includes mitosis, prophase, prometaphase, metaphase, anaphase, telophase. And cytokinases, cytoplasm splits into two. It has G1 and G0 are of variable duration. Regulation of cell cycle. Cyclin-dependent kinases constitutively expressed but inactive when not bound to cyclin. Cyclins, regulatory proteins that control cell cycle events. Phase specific, activate CDKs. Cyclin CDK complexes phosphorylate other proteins to coordinate cell cycle progression, must be activated and inactivated at appropriate times for cell cycle to progress. Tumor suppressor. P53, that is P21 induction, that will lead to CDK inhibition and hence RB hypophosphorylation activation, and that will go as G1 S phase progression inhibition. Mutations in tumor suppressor genes can result in unrestrained cell division. Example, Li-Fraumeni syndrome. Growth factors, example, insulin, PDGF, EPO, and EGF bind thyroxine kinase receptor to transition the cell from G1 to S phase. Here you can see all the cycles. The M, mitosis, cytokinases, and then growth, G1 as like GG, and then you have S phase where you have DNA synthesis, that is S, and then G2. This is the cell cycle. You can it's a diagrammatic representation of whatever we are talking over here. Cell types. Permanent, remain in G0, regenerate from stem cells. These are neurons, skeletal and cardiac muscle, RBCs. Stable, quiescent, enter G1 from G0 when stimulated, like hepatocytes, lymphocytes, PCT, and periosteal cells. Labile are never go to G0, divide rapidly with a short G1, most affected by chemotherapy. So bone marrow, gut epithelium, skin, hair follicles, germ cells. Rough endoplasmic reticulum, site of synthesis of secretory exported proteins and of N-linked oligosaccharide addition to lysosomal and other proteins. Nil's body, RER in neurons, synthesize peptide neurotransmitters for secretion. Free ribosomes unattached to any membrane, site of synthesis of cytosolic, peroxisomal, and mitochondrial proteins. These are mucus-secreting goblet cells of small intestine, and antibody-secreting plasma cells are rich in rough endoplasmic reticulum. Proteins with organelles, example ER or Golgi bodies, lysosomes are formed in RER. Smooth endoplasmic reticulum, site of steroid synthesis and detoxification of drugs and poisons. Lacks surface ribosomes. Location of G6P, which is glucose 6 phosphate, last step of glycolysis. Liver hepatocytes and steroid hormone producing cells of adrenal cortex and gonads are rich in smooth endoplasmic reticulum. Cell trafficking. Here you see Golgi is a distribution center for proteins and lipids from ER to vesicles and plasma membrane. Post-translational events in Golgi include modifying N-oligosaccharides on asparagine, adding O-oligosaccharides on serine and thionine, and adding mannose 6-phosphate to proteins for lysosomal trafficking. Endosomes are sorting centers for material from outside the cell or from the Golgi, sending it to lysosomes for destruction or back to the membrane Golgi for further use. One cell disease, number one, eye cell disease, inclusion cell disease, mucolipidosis type 2, inherited liposomal storage disorder, autosomal recessive, defective in N-acetylglucose amylene 1 phosphotransferase. This will lead to failure of Golgi to phosphorylate mannose residues, hence causing decrease in mannose 6-phosphate on glycoproteins, leading to protein which will cause proteins are secreted extracellularly rather than delivered to lysosomes. Result in coarse facial features, gingival hyperplasia, clouded cornea, restricted joint movement, claw hand deformity, kyphoscoliosis, and high plasma levels of lysosomal enzyme, often fatal in childhood. Signal recognition particle. Abandoned cytosolic ribonucleoprotein that traffics polypeptide ribosome complex from the cytosol to RER, absent or dysfunctional SRP, accumulation of protein in cytosol, vestibular trafficking protein, COP1, Golgi, which will cause Golgi retrograde, cis Golgi leading to into the ER. Where is CP1? This one, CP1 over here, and then CP2, COP2. COP2 in ER, which will cause cis Golgi enterograde. 2 COP2 step forward enterograde, 1 COP1 step backward retrograde. So basically they are trying to say that they're showing the movement of the COP and CO2. So CO2 is like 2 step forward and COP1 is 1 step back. Clatrin is trans group Golgi, that is lysosomes, plasma membrane, lead to endosomes, receptor mediated endocytosis, example LDL receptor activity. Here is the diagrammatic representation of whatever we have talked about cell trafficking. Peroxisome is a membrane enclosed organelle involved in beta oxidation of very long chain fatty acid, VLCFA, strictly peroxisomal process, alpha oxidation of branched chain fatty acid, strictly peroxisomal process, catabolism of amino acid and ethanol, synthesis of cholesterol, bile acids, and plasmalogens, which are important membrane phospholipids, especially in white matter of brain. Zellweger syndrome is an autosomal recessive disorder of peroxisome biogenesis due to mutated PEX gene. Hypotonia, seizure, hepatomegaly, and early death can be seen in this syndrome. Refsum disease, which is an autosomal recessive disorder of alpha oxidation, which here you see phytanic acid not metabolized to pristanic acid. This will lead to scaly skin, ataxia, cataract, night blindness, shortening of fourth toe, epiphyseal dysplasia. Treatment is diet and plasmapheresis. Adrenoleukodystrophy, X-linked recessive disorder of beta oxidation due to mutation in ABCD1 gene. VLCFA build up in adrenal glands, white leuco matter of brain and testes. Progressive disease that can lead to adrenal gland crisis, coma, and death. Proteasome are barrel-shaped protein complex that degrades damaged or ubiquitin-tagged proteins. Defects in the ubiquitin proteasome system have been implicated in some cases of Parkinson's disease. Cytoskeletal elements are a network of protein fibers within the cytoplasm that supports cell structure, cell and organelle movement, and cell division. Types of filament are microfilament, intermediate filament, and microtubules. Microfilaments are seen in predominant function is muscle contraction, cytokinases, example actin and microvilli. Intermediate filaments are function are maintain cell structure, seen in vimentin, desmin, cytokeratin, lamins, glial fibrillary acidic protein, GFAP, and neurofilaments. Microtubules. Microtubules are for movement and cell division, like cilia, flagella, mitotic spindles, axonal trafficking, and centrioles. More about microtubules. Microtubules are cylindrical outer structure composed of helical array of polarized heterodimers of alpha and beta tubulin. Each dimer has two GTP bound incorporated into flagella, cilia, mitotic spindles, grows slowly, collapses quickly. Also involved in slow exoplasmic transport in neurons. Molecular motor proteins is like transporter cellular cargo toward opposite end of microtubule. Retrograde to microtubule, that is positive to negative, dynein, and anterograde to microtubule is negative to positive, that is kinesin. So ready attack. It's kind of a mnemonic to remember. Clostridium tetani tetani, herpes simplex virus, poliovirus, and rabies virus use dynein for retrograde transport to the neural cell body. Drugs that can act on microtubules, microtubule M tubules, get constructed very poorly. So, as we also said, that they are very poorly bound. Mebendazole, antihelmintic, griseofulvin is antifungal, colchicine is anti-gout, vincristine is vinblastine, like anti-cancer, and paclitaxel is anti-cancer. Negative N near nucleus, positive N points to periphery. It's like a mnemonic to remember the retrograde and anterograde. Ready, attack, or negative N near nucleus, and positive N points to periphery. Cilia structure: 9 doublet plus 2 singlet arrangement of microtubules. We talked about microtubules. Here we are talking more about how the cilia are formed. Basal body, base of cilium below cell membrane consists of 9 microtubule triplets. We can see here 9 microtubule triplets with no central microtubule, nothing in between. Exonermal dynein is ATPase that links peripherally. 9 doublets and causes binding of cilium by differential sliding of doublets. Gap junctions enable coordinated cilia movement. Kartagener syndrome, which is primary ciliary dyskinesia. Immotile cilia due to a dynein arm defect. Autosomal recessive result in decreased male and female fertility due to immotile sperm and dysfunctional fallopian tube cilia respectively. Increased risk of ectopic pregnancy can cause bronchiectasis, recurrent sinusitis, chronic ear infection, conductive hearing loss, and situs inversus. Example, dextrocardia or on chest X-ray. Decreased nasal nitric oxide used as screening test. Kartagener's restaurant, take out only, there's no dine-in. Okay. Kartagener's restaurant, take out only, there is no dine-in. You get the joke. Sodium-potassium pump. Sodium-potassium ATPase is located in the plasma membrane with ATP site on cytosolic site. So here you have the 3 sodium goes out and by exchanging ATP to ADP and 2 potassium comes in. It's a trade. For each ATP consumed, 3 sodium leaves the cell, pump phosphorylated, and 2 potassium enter the cell, pump dephosphorylated. Plasma membrane is a asymmetric lipid bilayer containing cholesterol, phospholipid, sphingolipid, glycolipid, and proteins. So here it's like pumpkin. So it's like pump potassium in. When a cardiac glycoside inhibits by binding to potassium site. Cardiac glycoside, digoxin and digitoxin directly inhibit sodium-potassium ATPase, which leads to indirect inhibition of sodium-calcium exchange, which will lead to increase in calcium, hence leading to increase in cardiac contractility. Collagens. Collagens are of 4 types. These are most abundant protein in the human body, extensively modified by post-translational modification, organize and strengthen extracellular matrix. Be so totally cool. Read books. Okay, we will just elaborate that. Type 1 is most common, 90%, bone made by osteoblasts, skin, tendon, dentin, fascia, cornea, late wound repair. Type 1 bone decrease production and osteogenesis imperfecta type 1. Type 2 is cartilage indulging hyaline, vitreous body, and nucleus pulposus. Type 2 is cartilage. Okay, you get that? Type 3 is reticulum, skin, blood vessels, uterus, fetal tissue, early wound repair. So type 3 is early and type 1 is late wound repair. Type 3 is deficient in the uncommon vascular type of Ehlers-Danlos syndrome, 3D. Type 4 is basement membrane, basal laminar lens. Type 4 under the floor. You have the type 4 under the floor and basement membrane. Defective in Alport syndrome, target by autoantibodies in good pasture syndrome. Collagen synthesis and structure. This is a diagrammatic representation of what we will just talk over here, which is written in words. So number 1 is synthesis. Translation of collagen alpha chain, pre-pro collagen, usually XY, X and Y are proline or lysine. Collagen in 1/3 glycine. Glycine content of collagen is less variable than that of lysine and proline. Hydroxyproline is used for lab qualification of collagen. Quantification of collagen. Hydroxylation of specific proline and lysine residues. Requires vitamin C. Deficiency will cause cavity. Glycosylation is glycosylation of pro-alpha chain hydroxylation residues and formation of pro-collagen via hydrogen and disulfide bonds. Triple helix of 3 collagen alpha chain. Problems forming triple helix, that is osteogenesis imperfecta. Exocytosis of pro-collagen into extracellular space. Proteolytic process. Cleavage of disulfide-rich terminal region of pro-collagen, that is insoluble tropocollagen. Cross-linking, reinforcement of many staggered tropocollagen molecules by covalent lysine, hydroxylysine cross-linking, which are by copper-containing lysyl oxidase to make collagen fibril. Problem with cross-linking will lead to Menkes disease.